Hello, folks, and welcome. We're just going to tell made it, or let's say most people made it. I invited everyone that was registered to the last session also to this one, and I see the count of attendees going up. And while we wait, we will invite our first speaker on screen. We are ready to go. Hi, Eva. Can you see and hear me?
Yes. Can you hear me okay? How's the volume?
Yeah. For me, it works. Okay. It's fine.
Excellent. So I'll go ahead and share my screen. I don't have a second window open, so I won't be able to see the chats. Can you see the screen okay? Hello? Pierre?
Yeah, sorry, you were a bit off and on for me, but it's my connection. Let me just quickly.
Okay.
Sometimes it helps if I open.
When I share, we're all finding ways to boost. Okay, so other people are saying that it's fine. Great.
Oh yeah, I see your screen now as well. Let me just focus that one, bring it on here. That looks good. Great. And I think we got most people on here now. So to prevent further delays, I would say we start. Is that okay? Then I would introduce you and you can take it from there.
Great.
Awesome. So hi again, everyone. Great, you're still with us. And I'm very happy to introduce Eva Dyer to you. Eva completed all of her degrees in electrical and computer engineering, including a PhD, master's, and bachelor's. The former two at Rice University and the later at University of Miami, and she's currently an assistant professor at the Wallace H. Coulter Department of Biomedical Engineering at the Georgia Institute of Technology and Emory University, where she runs the Neural Data Science Lab, where she and her team develop new machine learning and data science approaches for making sense of large-scale neural networks. We're all super excited for your talk, and please take it from here, Eva.
All right, great. Thank you so much for the invitation. We were originally going to be in Montreal at this point, so I'm a little sad that we can't all be together, but I'm very excited that I have on unicorn slippers. So I haven't ever given a talk in such comfort. So yeah, so I'll go ahead and get started. My talk today is about work that we've been doing in basically taking neural recording. So I'll be talking about some kind of similar topics to what Carson talked about last time. So I'm really pleased that she did a great job of kind of setting the stage for a lot of the topics that I'll be discussing. So yeah, we're talking about, you know, taking neural recordings and being able to compare them or think about how they might vary or change across different types of perturbations to the nervous system. So whether it be across time, across space, so different subsets of neurons is what I mean in this case, or across different behaviors as well. So a lot of the work in my lab is motivated from the sort of emergence of many new techniques for recording high dimensional and very high resolution neural activity and also brain structure. A lot of this has kind of come to be over the past decade or so with the Brain Initiative, and these major advances have produced really high resolution and high dimensional data, much like what Carson was talking about in the previous talk, with these larger data sets, right, where we either have recordings from more neurons or where we have higher resolution of images that we've used to capture brain structure. In all these cases, we need to think of ways to actually make sense of all this data. And so my lab works on a number of different strategies for taking high dimensional data and making sense of it. And often that does involve finding lower dimensional formats to put the data into. So a question we are often faced with is why reduce dimensionality? And so there are a number of different rationales, different reasons depending on the application. In one case, obviously if we take something that consists of a lot of information, right, a lot of bits, a lot of neurons, or a lot of time points, and we can boil it down into a smaller set of numbers, then this allows us to effectively compress the data. And this is, the kind of core backbone of MP3 or any sort of compression algorithm, is actually finding a new basis or a new representation of the data that allows it to be re-represented in a lower dimensional or compressed format. In addition to compression, which is a kind of obvious one, there's also advantages of reducing dimensionality in terms of denoising. So essentially the idea is that if you know the data has sort of low dimensional structure to it, then when noise is corrupting it, this will often kind of move it off of this low dimensional subspace. And so by enforcing this low dimensional structure, we can actually get rid of a lot of noise or artifacts potentially. And then the third reason why we'd like to reduce dimensionality is often for interpretation of complex data. And I think that Carson did a really great job of setting the stage for that. Ultimately, we're faced with a question of whether or not we can understand what these complex systems are doing. And oftentimes when we refactorize or represent our data in a lower dimensional space, in some cases, we can actually place meaning or interpretation to those lower dimensional representations. So just to sort of set the stage for the rest of the talk so that we're on the same page, I'll be sort of thinking about general tools for dimensionality reduction, or many of the things I'll describe could be applied to different types of data, which, generally speaking would be represented, let's say, as a matrix, where we have a number of different features, and then we have different samples, in this case, along the columns. And so in the examples that I'll show right after this, and the sort of core motivating example, we're also thinking about a similar set of features as Carson's talk. So now each of the different rows essentially is a different neuron. And now we're tracing out the neural activity of each of these neurons over time. And that's essentially one example of this matrix that I'm describing here on the left. And now what we can think about is essentially representing now each column of this matrix, which is a point in time, and that represents the activity of, let's just say, D different neurons within our system. We can essentially represent that now as a point in this high dimensional space. Here I'm showing it in, a sort of three-dimensional view, but this is actually a higher dimensional space, but it's hard to, of course, visualize. So now each of these time points is now a point in this high dimensional space. And the key insight in low dimensional models and dimensionality reduction in general is that natural signals tend to not occupy every possible position within this high dimensional space, but oftentimes these data are sort of clustered or might live on these subsets within the larger D dimensions. And so one way to represent that is, I've already said the word, but is this idea of a subspace, which is what I'm showing here on the bottom, which is essentially fitting a linear or a planar surface to this scatter of all these different points in the high dimensions, right? So when we do things like principal components analysis or other types of linear matrix factorization techniques, we're essentially finding this linear approximation to this collection of data. Of course, the same idea can be extended to when data sets have nonlinear structure in them. So instead of having, this piece of paper or a plane, we can actually sort of fold this nonlinear structure around the cloud of points as well. And in this case, the term for this are manifolds. So I've written here manifold models. That sort of refers to these nonlinear subspaces of the space, of the larger space. All right, and there's many different extensions of these sorts of ideas depending on the sorts of low-dimensional structures that one might expect to find within their data. I'm sure many of us are very familiar with the idea of clustering data, and we can also think about, if we look over here on the bottom right, looking at this cluster model, we can also think about it as itself a low-dimensional model where we're essentially representing this collection of points now with their centroids. And so this gives us a form of sort of compression and re-representation. And on the left, I'm sort of just showing a hybrid of all of this, where we would consider a case where we actually, instead of having one subspace or one manifold, we might have a mixture of these subspaces or manifolds. And I'm describing this model because we'll see that it becomes useful in our later discussion, this union of subspaces model. All right. If there's any questions at this point, I don't know, Pierre, just, I guess, chime in and let me know if there are any questions in the chat. Okay.
So far, we have none. I will let you know once something comes up.
All right, cool. So yeah, so we sort of set the stage for this. The sort of general idea is, we take recordings, in this case of neural data, we find some low-dimensional representation of it with a linear or a nonlinear technique. And then when we have this low-dimensional or representation in this latent space, so to speak, we're often now faced with the sort of next question, which is, how can we take these two different distributions of activity or distributions in this latent space, and how might we be able to compare them across, say, two different data sets? So this arises in many different neuroscientific applications, and of course in many other areas of machine learning. Some examples are, I have a collection of data, before disease onset and then after, for instance. That would be a comparison. We could also consider the case where, the first distribution is from a single time point and the other distribution is now from a subsequent point in time. So in all of these cases, we're often faced with this situation where we've gone through the process of finding a low-dimensional representation of our data, and now we're interested in how these two data sets are actually related to one another. And in many cases, while PCA is one example of a matrix factorization technique that is unique, the actual representation of these data in this low-dimensional space is not guaranteed to be the same. So what I mean by that is that, in many cases, we might find something similar to what I'm showing here, where we have a rotation or where we have some affine transformation of an original data set, and then what we see in the next data set is, a transformation of the original. And so in this case, we'd like to ask the question of how can we actually figure out a transformation that would align these two different distributions or two data sets, and that will then allow us to tell more concretely about how they've changed or how they've been altered across these different states. So really the goal here then is to figure out this function that will, bring these two data sets or distributions into alignment. So we can actually write this out or formalize this sort of idea with the following problem statement that I have here, which we'll refer to as a distribution alignment problem. So essentially what this is saying, so on the bottom I show here sort of visual for the high level strategy, which is we don't know the color of all these points, right? We don't know that the red correspond to the red and the blue correspond to the blue. We just have the shape of these distributions in this case. So what we'd like to do is be able to find a way to essentially, you know, figure out that the blue and the blue should be connected and the red and the red should be connected to one another, or are corresponding points within these two data sets. And so the formulation that we have here on top is essentially we have mu, which is our source distribution. And I'm sorry I don't have a pointer. I wish I could kind of interact with this a bit more. But so we have mu, which would be our source distribution. We have nu, which is our target distribution. We're going to apply a transformation t to mu in order to align it with the target nu under some distance, which I've written here as math or subscript d, right? Or this divergence metric d. And we're also going to constrain the possible class of transformations to lie in a set T in this case. So this is a very general problem statement at this point. Hopefully it's clear. Our idea is to learn a T that brings mu and nu into alignment with one another. So there are kind of two main design variables in this problem here that we need to think about. The first is, you know, which class of transformations we should consider. Obviously, considering all possible transformations makes this very difficult. In addition to the class of transformations, we're also faced with a question of which distance we should use to actually form or solve this alignment problem. And so to answer the second question about the distance, we've chosen a distance that comes from the literature on optimal transport. It's called the Wasserstein distance. I'll explain to you a little more in the application why the Wasserstein or why optimal transport distances are favorable, not just for this problem, but for sort of general transfer learning problems more broadly. But just to provide a little bit of intuition about what it actually is. So, you know, many of you, I'm sure, are familiar just with the normal notion of a norm, or sort of the Euclidean distance between 2 points. And so if we consider here now the distance between these three different distributions here on the bottom, we have a blue, a purple, and a red. So because these three distributions are perfectly non-overlapping, then if we measured their distance in a normal sort of Euclidean norm or any other norm, we would say that they're all actually orthogonal to one another, right? Because they don't share any overlapping support. And that's sort of shown here with this triangle on the left, denoted LP, just that all of these distributions are equally far from one another. However, if we sort of think that the position in space is actually important for telling us information about nearness or closeness between these distributions, then we actually would want to potentially say that the blue and the purple are closer to one another than, say, the blue and the red, which are much further away from each other in terms of this axis, or in terms of the support that we're showing here. And so this sort of notion of geometry of closeness can be captured by the Wasserstein distance. And that's what I'm showing over here on the right, which is that the blue and the purple now are much closer to each other in terms of their Wasserstein distance than, say, the blue and the red. All right, so we can kind of break this down a little bit further. So this is just the definition of the Wasserstein distance, and in particular an entropically regularized version, which makes the complexity much better, and basically convergence is much quicker as a result. So here what I'm showing is just the the definition of the optimal transport distance. So what you see is that it feels very complicated, because now all of a sudden, to even measure the distance between these two distributions, I actually have to solve an optimization program. But as you'll see, when you use this entropically regularized version, the updates are actually quite simple, and so this can be computed efficiently. So I'll just kind of step through what it is, because I think there's some nice intuition here. So essentially, your Wasserstein distance between two distributions, P and Q, is given by basically the minimizer of this program. So what we're doing is we're looking for a matrix P, which tells us how we're going to transport mass from distribution P to distribution Q. So P is basically the main operator that tells us how these two distributions are related to each other. And we're going to essentially multiply the entries of this transport matrix by the cost to transport between those two different bins. So we essentially give a cost associated with moving mass from bin I to bin J, and we're going to penalize now that transport based upon that distance. And in our case, we can just use the Euclidean distance in space as our sort of cost. So that means that when we have to move much further away in space, we're going to pay a higher cost. And then what we see here on the right is this entropy regularized term. And so this will essentially help to smooth out our problem, and we can think about it as a regularization term that also can in some ways be thought of as a way to help avoid overfitting in some cases. Okay, we need to make sure this is a valid transport plan, and so these are the constraints essentially to make sure that this is a doubly stochastic matrix. And then here, this is the entropy term, just writing it out. And now this just provides a little bit of visualization, or hopefully some intuition about what this entropic regularization does. So essentially what we're showing here on the left is if I'm trying to transport mass from P to Q, right, and they're shown along the rows and the columns, the optimal transport, or the transport plan with no entropic regularization, is shown on the left. And then as we increase the amount of entropic regularization, as we move to the right, what we see is that basically the transport plan is being smoothed. And that smoothing is actually very beneficial in terms of solving this efficiently. All right, so just recall, going back to our original alignment problem, which we see here on top, we had these two questions. One was, which types of transformations should we consider? The other, distance. So far, I hope I've convinced you, at least at this stage, that optimal transport can make sense when you have a geometric sort of, some geometry underlying the sort of space, right, over which you would want to compute distances. And then the second question is, which class of transformation should we consider? And so this in general is a hard problem, and it's very data set dependent often, and much of the field of transfer learning, which tackles these sorts of problems, is very specific depending on the types of shift across different data sets. And so we will then turn to our neuroscience application to really help us constrain the space over which we're going to delete. I guess this would be a good point to stop if there was any questions about the math or the sort of methods up until now. Okay. Great. So then, yes, so let's turn to our application. So in this case, what we're looking at is a situation where we can, implant an electrode array and measure the activity of individual neurons within different parts of the cortex. In this case, we're looking at primary motor cortex, which is, involved obviously in coding movements. And so we're recording neural activity while we're also monitoring the motion of the the individual's reaching movements. So basically there's a queue for the individual to make a reaching arm movement to one of 8 different reach directions, and then there's a center point. So this is a kind of standard center outreaching task, as it's called. So what we're doing is we're able to both see neural activity simultaneously while we can actually monitor the kinematics or the velocity of those different reaches into different directions in space. So we have a 2-dimensional behavioral readout along with the neural activity. And a sort of general question that we're interested in with these sorts of data sets is what's called neural decoding. So basically finding a mapping between neural activity and the movement. And traditionally, this is done by, recording both of these things simultaneously, finding some mapping between the two, say on training data, and then using that, on a test data set after you've learned that relationship on the previous data. So in this case, what we can actually do is we can, and in some of our previous work, we've shown that you can take the activity of these neurons in motor cortex, right? You can reduce them down to a lower dimensional space, which is what I'm showing here on the bottom right. So this is after applying a linear dimensionality reduction technique, fact analysis, to embed the data into 3 dimensions in this case. And so we know that without any labels, without any correspondence between the neural activity and the movements, we know that the shape of the movements are actually preserved in the neural activity. So we were interested in using this sort of alignment method that I've described to think about how we can bring this high dimensional, at some point, neural activity down to a low dimensional space and then align it with this movement distribution, which I'm showing here on the left. And if we can do that effectively, we're able to essentially solve this neural decoding problem that I've described, but to do it without needing correspondence between the movement and the neural activity. This would then allow someone to potentially decode from a subject that they might not have captured training data from directly, but they know that the subject is actually doing the same underlying task that should have the same representational structure. All right. So based upon this sort of observation and the motivation of our problem, where in this case we have structure within the neural data that has clusters, right? Or this sort of union of subspaces model that I referred to earlier. We have that activity of neural populations when the subject is moving towards a particular reach direction tend to be clustered in this latent or low dimensional space. And based upon that, we basically have this clustered representation that we can exploit within our formulation. So I'm running time. I spent a lot of time going into details on OT. So I might kind of move quickly through some of this, but essentially the idea just at a high level, if you take our original optimal transport alignment problem, and now we're going to split it into essentially trying to find across clusters as well as find correspondence across individual points. And so by breaking this in this way and solving it in these 2 different stages, it both effectively aligns with less data and also avoids a lot of the local minima within this problem. And that's why I call it hierarchical, because it's sort of this two-tier approach. And so what I'm showing here is just the new formulation, similar to what we've shown before, but now we've essentially split our Wasserstein divergence, but now over these transformations of the individual clusters. So I'm just showing here, the breaking down of the source and the target into clusters. And now our objective is now going to be aggregated over all of these different alignments. And finally, in the end, we can enforce constraint, a constraint that all of the TIJs, so all of the individual transformations, have to agree on a global transformation. And that's just one assumption of our model. But one might also potentially extend this to consider a case where you could have different transformations for for different clusters potentially, if that's how you expect your data to be shifted. Here, we're also constraining T to be the class of unitary transformations. So in this case, we can find basically a rotation of our data that helps to bring these things into alignment. So the key contributions of this work, which was a paper at this past NeurIPS, was this hierarchical formulation of OT. We also have a distributed solver. We have both MATLAB and Python implementations if you're interested in testing it out. And we also show improved performance in neural distribution alignment. This is just some results on synthetic data showing you that HIWA, which is our method, hierarchical Wasserstein alignment, which is the blue on top, provides major benefits over just a simple Wasserstein alignment. So meaning if you weren't to leverage this clustered structure within the data. What's nice is that this, in a synthetic 2D case, we can show that using optimal transport over just a KL divergence, which was the original divergence measure that we used here, really smooths the landscape, and it reduces a lot of the local minima that the algorithm can get trapped in. So this is a really important part of the solution, both using optimal transport as a law, and also leveraging clustering structure when it does exist within your data. These are some examples and results of now trying this on 2 different non-human primates. So on the left, I show, one, and on the right is another. Here, because both of these individuals are carrying out the same motor task, which is constraining their neural activity to be low dimensional and have similar structure, we can actually now take neural activity from one animal that we haven't actually formed any target data on, or we don't have the movement. We can align it onto a target data set from a different individual. And so in this case, given that the constraint on behavior is consistent, we can actually find a cross-subject alignment in this case. This is showing that this is robust to subsampling. So within the macaque motor cortex, we find that, even though we have 150 neurons, we don't actually need that many to be able to decode reliably. And in some cases, when good subsets of neurons are selected, then you can do a lot better. And so maybe we can use Carson's method to find the good sets of neurons. We have also now extended this to rodent cortex. So in this case, this is in striatum, a collaboration with Keith Hengen's lab at WashU in St. Louis. Here we're showing now 2 different data sets that are acquired over 2 different 12-hour blocks. And in this case, we see over periods of the animal being awake or being in 2 different stages of sleep, we actually find very consistent and reliable sort of geometry or shape across these low-dimensional embeddings. And this allows us to then align these 2 and provide, a good way to then bring these data sets back so that any labels that you've collected in terms of sleep on a previous data set can then be transferred onto this new one. And we compare here with canonical correlations analysis as well. So I know we're running out of time, so we can play similar games here. Robustness to subsampling is lesser. We also have fewer neurons. So we're sort of interested in looking at how all of these methods apply when you're, obviously not able to acquire the number of neurons that, might be involved in this full circuit. And so I think that this is, I have, just 5 minutes left, right? So I think this is a good place to stop. Actually, okay, I'm just going to give you a little teaser. I wanted to talk about this. And I just wanted to quickly tell you about this other story, but it's related in the sense that, there are many cases where you might not already have a good feature space to work with. So in our previous case, we were able to take individual neurons over time and use that as the basis for solving this problem. But in many cases, especially when you're dealing with images, you might not already have a good feature space. And so deep learning has emerged as a powerful technique for feature extraction and representation learning within high-dimensional image data. And I just wanted to tell you quickly about some work that we're doing where we call it deep brain disco for deep brain discovery. Essentially, the idea is to start by training a neural network to discriminate different brain areas using small local snapshots of the underlying anatomy. We train the neural network to solve this discrimination task, so pretty standard convolutional neural network. And then what we go in is we actually go into hidden layers within the network and use now the network as a feature extractor to tell us more about the data than what it's actually been trained to do. So we use the network as a feature extractor and then use dimensionality reduction on top of that to provide interpretable views into the underlying microarchitecture and anatomy within this data set. And we can use that to start to discover some interesting features of the data. So on the right, I'm just showing examples of manually annotated layers within our data and how our network is able to actually pull out these different lemma differences and start to group different regions within our sample that it hasn't been trained to do because it's essentially learned a rich set of features that describe the underlying structure in the data. And we can also pull out some fun things like, finding high density, high cellular density regions also without training the network to do so. All right, so in summary, I told you about work that we've been doing using dimensionality reduction and also this optimal transport alignment technique to be able to do things like movement decoding in non-human primates and also decoding sleep and wake within rodent. And then at the end, I told you about some work for in using deep learning to learn representations to then find patterns and discovery in neuroimaging data. And we also have a new X-ray micro CT data set along with that for the community, along with annotations to help with other people developing machine learning methods on sorts of data. All right, and of course, the team that made it happen. So the main core work has been done by a student in my lab, Beja, as well as John Lee, also at Georgia Tech. And then the later work was Aish Balwani in my lab. And then we have amazing collaborators. I talked about some data from Keith's lab and our funding sources. And with that, I will end the presentation and take any questions. Thank you.
Thank you very much, Eva, for this very great and informative talk. I guess there's nothing like embeddings, dimensions, and alignments on a sunny Friday. There was a very great explanations as well. Really enjoyed it, though. I searched for a disco emoji, but there unfortunately is none. So please just think about it. I was about to reply for your disco, deep brain disco thing.
I had one on my slide.
Oh,
I had one. I'll share it with you. Yeah.
So we have two questions I think we can just briefly address. It wouldn't be that bad if we go 2 minutes over time. We have one question from Roberto asking, is this method suitable for generalization across subjects? Does it need a high number of data points?
Yeah, that's a great question. So, I did talk a little bit about this result, right, where we could actually show a cross-subject alignment, taking a movement data set from one non-human primate and now taking a neural activity from a completely different subject, right, and finding a way to align the two. But of course, that relies heavily on the sort of task structure being sufficient to sort of constrain these neural activity patterns to be similar, right? So, in the case of the types of transformations that we use, right, we are assuming certain sorts of transformations, namely that they're not skewing or stretching the data. So, if I imagine that in a lot of other application settings across subjects, if you don't have enough to constrain them to be similar enough, you're going to see sort of a shift across domains. And in that case, incorporating those additional constraints could be added into this sort of OT framework. But currently, we don't incorporate kind of more sophisticated types of transfer.
Great, thanks a lot. And we have a second question.
Yeah, I see here. OT works with statistical distributions. How do you go from brain data to distributions? So, so what I've shown here is essentially taking, you know, neural activity by time. And what we first do is we find a low dimensional representation of that data using something like PCA, or if it's nonlinear, using isomap, for instance. And so the method that I'm describing is working directly in the latent space. And now in the latent space, you're essentially, let's just say it's a 3-dimensional space. You now have the probability that you're going to have mass at a particular position in the 3-dimensional space. And the key idea is that, you know, the first dimension for data set 1 might be a different set of dimensions, right? And so even though we have good representations of these 2 data sets in the low dimensions, we might need to now find correspondence across their PCs. Okay. Hopefully that answers the question. I think maybe the internet connection was not great.
Yeah, I mean, that's the beauty of it. Like, we can stream it on YouTube, or it will be streamed in parallel on YouTube. Then we're going to upload the videos after cutting them to talk-specific sections. So if anyone, like me, for example, experienced connection problems, you can check it out later on the YouTube channel. So with that, I think we would call it your talk right now. Thank you very much again, Eva, for this amazing presentation. And
yeah, thank you for the invite.
It was our pleasure, really, though. And now we're gonna head over to the next session. That is, I'm closing this down. I'm gonna invite you over to see all of you there in a bit.
like coming from the group of people who sort of define what the official weight of a kilogram is and that kind of thing. So reproducibility is defined as the similarity of results of the same or similar measurements. A measurand is like the thing being measured, carried out under changed conditions of measurement. So the thing about reproducibility is that you have to specify what are the changed conditions of measurement, and that is the reproducibility that you're seeing. So for example, if we have changed conditions of measurement across different scans, then that's between scan reproducibility. If we have different sessions, so take person out of the scanner, right, and then they come back later, maybe another day, then that's between session reproducibility. If we have between sample, then that might be two groups of people, and we look at what the parcellations look like between those two groups of people if we were to conduct a functional parcellation on both groups and then assess the similarity of those functional parcellations. And then we have reproducibility across studies. So if we have one group of one study over here with a set of functional data and another study over here with a different set of functional data, how similar are those two studies in terms of their functional parcellation? And then finally, populations being like, are the underlying sort of assumptions about the type of people who are being scanned different in some important way? So there are a lot of different applications of bootstrap aggregation in the literature, most of which focus on methods that can assist in prediction. So you might be most familiar with bootstrap aggregation in the context of random forest or something like that, right? It's a set of ensemble methods. And there's an element that we think would be applicable to functional parcellation and bagging, in that there's an important element that the sample that you ended up getting, right? Not only the sample in terms of the subjects, but also the sample in terms of the time series that you ended up sampling are random. So the people who ended your study and the people who ended up or the hour of the day or what they were thinking about at that exact moment are somehow random, right? And so we don't want to, when we're conducting a parcellation, to be actually saying that this clustering is we want this clustering to accurately represent exactly what was happening in these people's heads at that exact moment in time. We want to somehow get an assessment of like the variability in both the sample in terms of the individual and in terms of the time series. And so this bagging approach, and I'm going to show it in a more concrete example in terms of neuroimaging, but generally works like this. So we have a data set and we create some kind of resampling step that enables us to create multiple data sets from that original data set, and we perform a clustering on each of those data sets. And then there's some kind of consensus aggregation across all of those different clusterings, which then gives us our final clustering in the end. So the first half on the left-hand side is our, I'm showing with my mouse, is the first half on the left-hand side is our bootstrapping, and the right-hand side is our aggregation, and that's where we get this bootstrap aggregation. And so PyBask is a Python-based implementation of an extension of this bootstrap analysis of stable clusters method that was developed by Pierre Belek and shown in 2010. And I'm going to briefly describe how this kind of approach works. But in order to do that, I'm going to first describe a sort of a more standard approach at how we might look at clustering. So for example, we have in the top here, the standard approach, we have different subjects, subject 1, 2 to subject n, and we take their pre-processed fMRI data for the whole brain, and then we represent it in some kind of super voxel resolution, right? So the brain has tens of thousands of voxels, and it's computationally very demanding to be able to actually perform clustering on such a high high large matrix. So we downsample it to something more like in the range of thousands instead of tens of thousands. And then from each of those supervoxels, we might extract, we would extract a time series. That's the average of all the voxels in that supervoxel. And the supervoxel can be created with a variety of different methods. People can use some kind of hierarchical feature agglomeration. That's what we do here with using Nilearn. And then you know, a time series is extracted, and for every supervoxel, and a clustering is applied, and then that clustering is represented here in this thing called a adjacency matrix. So this adjacency matrix is a supervoxel by supervoxel sized matrix, which has a 1 if two voxels are in the same cluster, or a 0, or sorry, if two supervoxels are in the same cluster, or a 0 if they are in a different cluster. And then those individual adjacency matrices all get aggregated together to create a final group adjacency matrix, which is then itself submitted to a final clustering to get sort of what the final parcellation might look like. In the case of bagging, there are two different steps that bagging is happening now. So if it was just more sort of standard bagging procedure, we would just be bagging across individuals. But since we have this, we have time series, and we think that we could probably show benefit from bagging the time series as well, we do it at this multi-level bagging approach. So the first step is, in the same regard, you take the individual's data, you create this supervoxel resolution, and then you use bootstrap aggregation, or bootstrapping rather, first to resample that subject's 4D time series into a set of, say, 100 or 200, or even as little as 5 or 10 other time series. So we take from one 4D matrix now to like X 4D matrices, where X is the number of bagging bootstraps that we're doing. And then each of those resampled 4D matrices are then, and I should just mention that the type of bootstrapping here that is done is called a circular block bootstrap. So it's taking contiguous pieces of the time series and concatenating them together. And the pieces that are taken are themselves chosen at random. And so we have these multiple time series, and then each of these 4D images gets clustered, and we get an adjacency matrix for each bootstrap. Okay, and then those adjacency matrices for this, still now only at the individual level, those adjacency matrices for the individual subject then all get aggregated together into this mean individual adjacency matrix, right? So whereas the adjacency matrices themselves, they are binary. They're ones and zeros. Ones where when two voxels got clustered together, zeros when they weren't clustered together. Now they get aggregated into this with this mean adjacency matrix. So now what we're seeing is the proportion. So each cell now represents the proportion of times across bootstraps that that two voxels were clustered together, right? So if we have 100, bootstraps, then we would have some value between like 0 and 100 that described how many times the data were clustered together. And then at this second stage, so I'll just point out that the red arrows all here show the bootstrapping part, and then the blue arrows show the aggregation part. So this next part here is, all these red arrows are showing the bootstrapping part. So now, but the bootstrapping part now at the group level. So now we're aggregating now we're bootstrapping not the time series, but now we're bootstrapping which of these mean adjacency matrices get included into a group, a bagged group adjacency matrix, right? So now we have different groups of individuals. So this one has some proportion or mixture of the individuals in our sample. This one has another proportion of individuals in our sample, et cetera. They all get aggregated together into this mean bagged group clustering. And then that final clustering is then, or that final adjacency matrix or stability matrix is then clustered to produce the final parcellation. All of this in PyBask is done with the help of Nilearn and scikit-learn and NiPipe, which are all really wonderful tools. I'm very happy to have gotten to use them. So a little bit about the samples that we use in this study to sort of assess whether we can really improve the reproducibility of functional parcellations using bagging. We used two data sets. One, the Hangzhou Normal University data set, which is a fantastic stellar data set if you are interested in reproducibility. It's 30 subjects. They are each scanned 10 times for 10 minutes per scan. They are all in their mid-20s and 50% male, 50% female. And we have the genome superstruct project. The HNU dataset was collected in China, in China, and the GSP dataset was collected at Harvard by Randy Buckner's group. And Avron Holmes is the first author on that. So, and Xinyan Zuo is the first author on the HNU core initiative paper. This GSP has 300 subjects. Each of them are scanned for 2 6-minute scans. So this is within session. So they have two scans in the same scanner session. Whereas in the HNU, it's 1 scan across 10 sessions. So this enables us to look at both between session effects and between scan effects. And the large sample of the GSP data set allows us to look at within population, within study, between sample effects, and compare actually these two. So I'm going to go over a little bit of different types of generalization that you can think about when you're when you're talking about reproducibility in this context, and that we studied specifically in this study. So first, and I put these little blue circles and numbers here so that you can kind of keep track as we go through the figures. You can kind of keep track of which level of reproducibility we're looking for. So generally, this 1 is the closest reproducibility, and then this 5 is the furthest away, right? So between scan, they're the same population, same sample, same scanner, but the data is different because it was just acquired, several minutes apart. So we have this GSP 10 group test retest, so the 2 scans. Then we have, for 2, we have this between session, which is same population, same scanner, same data again, but now we're looking at the HNU because now we have 2 sessions, or at least 2 sessions per person. So we have 10 per person actually here. And we have reliability, where we are actually looking at the relationship between the variance within an individual across sessions and scans, and to the variation between individuals. And then down here at 4, we have within study between samples. So this one's kind of a mouthful. So within study means that we're looking at the same study. So we're still looking at the GSP, but we have between samples. So we have 300 subjects. So now we've split those 300 subjects into 10 groups of 30, right? To keep them consistent with the size of the HNU data set. And so now we have the same population, but now different sample, different scanners, and different data that are acquired because the GSP was acquired in multiple scanners. And then finally, we have between study, between sample. And this is sort of the furthest level of reproducibility that you can imagine. And ideally, the one that you want to, that one that we are hoping for the most, right? Because if we can't get, if we have between study and between sample reproducibility, then we feel pretty confident that we can sort of say that this method would help harmonize results across different data sets, not just different data sets that are acquired within a single study. So the approach that we took here was we looked at both the question of cortical parcellation and subcortical parcellation, both in parallel, because while cortical parcellation sort of gets the sort of majority of attention and effort, the subcortical parcellation is also really important, and is actually a particular interest of mine. Subcortical areas and cortical striatal interactions are a particular area I'm interested in, and they also are an area of the brain that have typically less, well, people think at least have less signal to noise compared to cortex. That's mostly because of head coils, but there also may be endogenous elements of how subcortical areas are connected to the rest of the connectome that drive variance in their connectivity and time series that make them look less reliable. So we wanted to sort of demonstrate that this parcellation reproducibility element would be consistent regardless of which area you were looking at. And then the sort of overall picture here is to look at the impact of bagging on parcellation reproducibility and how that effective bagging interacts with the length of the scan time. So how much scan time do you have in your scan? What parcellation target, where are you parcellating, and which number of how many bootstrap aggregations are you actually using? Because as you use more and more bootstrap aggregations, it becomes more computationally demanding. So the first result, and I guess I should maybe pause for just in case there are questions. I'm not seeing any. Are there questions? I see. Gael, should we believe that there are indeed functionally homogeneous regions of an individual brain given the complexity of the brain? No, I don't really think that there are, but that's not going to stop people from trying to analyze the brain. I think there are, we can say something like this. There may, given that a lot of people, and I'm going to talk about this sort of at the end as well, but given that a lot of people are focused on discussing the sort of variability in the time series and variability of connectivity, we shouldn't necessarily believe that there's anything like a strict atlas in terms of what a region of the brain should really be. But there may be some kind of central tendencies that may exist. And personally, I tend to believe that perhaps rather than maybe looking at from this perspective of the entire brain, maybe breaking it up into sort of smaller interregional parcellations or between network parcellations might be more realistic. For example, we might not be able to understand what would be the best approach to parcellate the, say, the paracingulate gyrus or the precuneus, but maybe with respect to how the default mode and the frontoparietal network are related, or the singular network are related, there may be some kind of more dominant consistencies there. Okay, going back to this. So, okay, so this is our sort of first stab approach. So we're looking now at between scan reproducibility. And so on the left-hand side, we're going to see a lot of these four panel figures. So I'm just going to use this one to sort of introduce how they're shaped. So on the top here, what we're looking at is the spatial correlation of those adjacency matrices. So how similar is the voxel by voxel relationship in terms of the parcellation. And on the left-hand side, we're always going to have the non-bagged condition. And then after the dotted blue line, we're going to see what it looks like with 10 bootstraps, 100, 200, and 400. So, and then generally we'll have for these, this one is subcortical data, and this is looking just at 6 minutes. So 6 minutes is like on the lower end of the amount of time that we ended up investigating. We go up to 50 minutes at the upper end. And then we have two different parcellation resolutions. We have K10, so 10 subcortical clusters, and then K18 is 18 subcortical clusters. And then this second metric down here is called the adjusted RAND index. And so this is a sort of a permutation free method of measuring how similar the voxel cluster assignments are. So for example, if I were to measure two parcellations, one with a cluster label of 1, 2, 1, and the other with a cluster label of 2, 1, 2, the adjusted RAND index, since it doesn't care about these permutations, it just matters about the overall pattern, it would see those as identical. That would get an adjusted RAND index of 1. So we see generally that bagging improves the between scan reproducibility of our parcellations. And then here we're taking a look at cortical parcellation. So this is 2. Now we're looking at between session reproducibility, and we're looking at the reproducibility from 7, 20, 197, and 444 clusters. I think one thing that we should note specifically is that generally we see a decrease in the consistency of our parcellations as we go into higher orders of regions. This is only based on 10 minutes of data. Again, no atlas that really people are using was created on 10 minutes of data, but this was sort of like a proof of concept, more or less, to demonstrate that even with 10 minutes of data, the bootstrap aggregation would improve. So we see generally that we are improving the reproducibility, and I think one notable thing to look at is, so here the red ones are the non-bootstrap condition, and the teal are the bootstrap condition. One thing worth noting is that the reproducibility at the level of sort of what we would typically call like a network, which is like a 7 parcel resolution, is like what we see as the highest, and we can and since as we go down into finer resolutions, the reproducibility also drops. It's a nice thing to see that the bagging at 197 clusters can give us something that can be about on par with what we would get with a 7 cluster resolution, suggesting that bagging can be one ingredient that can help us investigate more finer resolution organization of the brain. So this is now looking at, so that the previous slide I showed you was with 10 minutes of data, and now we're looking at a range of different amounts of data. So from 3 minutes, 5 minutes, 10 minutes, 20 minutes, 20. And we're showing that same pattern here. So now this is looking at the subcortical parcellation. In particular, another reason that we did this cortical and subcortical parcellation was that that was pretty nice, was that since the subcortical parcellation was so much smaller, we could run many more conditions with the same amount of cost on Amazon. So it enabled us to like explore the search space of the effect of bagging more broadly. So we show for spatial correlation, we show that if you look at that adjacency matrix, even 3 minutes of data shows very dramatic improvement in terms of reproducibility, even with only 10 bootstraps, right? So as we go into higher and higher bootstraps, we see, and we get up to 50 minutes of data, we almost see a perfect correlation between the two, between multiple sessions. In the case of, there are 10 scans here that are spaced apart on an average of 3 days. To create the 50-minute scan, what we did is we aggregated all of the odd sessions, and then to create the reference, we aggregated all of the even sessions. And all of these 3-minute, 5-minute, 10-minute, et cetera, they're all compared against that same 50-minute reference that we're calling our gold standard, right? So as you see, as you get into higher amounts of data, you get almost a perfect alignment. And we see that this pattern is the case for the spatial correlation for both K10 and K18. And the adjusted rand index shows a similar story, except that it doesn't actually seem to improve the parcellation at the very lowest amounts of data. So for 3 and 5 minutes, we don't see as much improvement as we do for, say, 10, 15, 20, et cetera. One thing that's really worth noting here also is that the bagging is improving the reproducibility of the parcellation more in some cases than doubling the scan time, right? So we can see, for example, going from 15 minutes to 30 minutes in the non-bagged condition leads to a smaller improvement than going from 15 minutes, going from 0 bootstraps to 400 or 100 bootstraps, for example, in 20 minutes. So if you look here, for example, you'll see that from 20 minutes to 50 minutes, we see this increase, but from this 20 minutes to this 20 minutes, we see a greater increase with bagging, right? So this is suggesting that bagging could be a way for us to get at more reproducible measurement with relatively less data. And this is showing like what those actual difference maps end up looking like in the 10 versus the 50-minute data. What we're tending to see is that it's often in areas of transition that we are sort of seeing the most difference, and these differences are quite reproducible across the different amounts of data. Now looking at the individual level, so now we take that, we have that group level parcellation, but now we're looking at our individual level parcellation as well. And we are seeing that we get better sort of parcellation at the individual level with more bootstraps. We see the same improvements in ICC as we would expect based on what the group level bootstrapping results are showing. We see improvements in discriminability. So discriminability is like a multivariate version of ICC. And we see improvements in discriminability as well, though those improvements of discriminability tend to be limited more to the higher number of minutes of data, 20, 25, 50 minutes. So discriminability means like how much can you pick out a person from your data. We see greater within study between sample reproducibility. So this is that those 300 subjects that are cut up into 10 groups. We see more similarity in the parcellation between the different groups there. This is in subcortical. We see also increases in between sample, between session reproducibility. So now this is looking at the cortical parcellation data set and comparing it to the cortical parcellation from the GSP data set and showing that if you use bootstrapping on both, you actually get much closer alignment overall than if you don't. This is only with 10 minutes of data. We see the same thing for the subcortical parcellation as well. The differences, interestingly, that we see here also are kind of similar to what we saw in the difference maps for the repeatability or the between session reproducibility. And the interesting thing is that we actually see a little dip in the reproducibility of the 50-minute data, and a lot of that has to do with the fact that with bagging, you actually start getting a better fit, a better parcellation. So the 50-minute parcellation ends up looking better than the 10-minute reference or the 10-minute that you were comparing it to. So we see that it seems like actually, though the reproducibility was it was still improved. It just wasn't showing quite as dramatic effects. It looks like it has to do mostly with the pattern of the 50-minute was looking actually more bilaterally symmetric than the 10-minute was. So the 10-minute, for example, is showing this caudate tail is not different between the two parcellations, whereas in the 50-minute, it was showing that the caudate tail on one side was different. So that's just an explanation of that. In the paper, we discussed this more. This is one thing that I think is particularly cool. I wasn't necessarily expecting, but we also found that bagging has really large improvements in the group to individual alignment. So if we look at the individual parcellation and the group level parcellation, we see that bagging shows really great effects in terms of improving how similar the individual parcellation and the group level parcellation is, which is exactly what you want, because if you were to conduct an individual level parcellation on your subject and you find it were to be really different from your group level, then you wouldn't want to use that group level atlas on that individual person. And then we also demonstrate that this replicates across other parameters, clustering parameters, such as spatial constraint, either with a small amount of spatial constraint or a fully spatially constrained clustering algorithm, and also with different parcellation techniques. So this is using spectral clustering, and we use hierarchical agglomerative clustering in the Ward's criterion base. In the actual study. I think I'm running out of time, but I just briefly want to talk about why bagging works, or why we think it does at least. So, supervised and unsupervised learning methods, they suffer from variability due to sampling, and the variability can also arise due to recursive dimensionality. So the idea is that aggregating across multiple resample data sets can provide you a more generalizable model. And because when you assess each of those individual each of the models are capturing different elements of the structure of the data. And so even if you were to develop some amazing algorithm that were to sort of select the best clustering or the best parcellation possible, you're throwing away some other information that are contained in these discarded non-optimal models, right? So like, each algorithm is parcellating, whether it's based on the data or the algorithm itself is changing. They're parcellating the data from slightly different perspectives. You can think about it, so you get a more holistic perspective. There's another element that perhaps beyond nuisance signals such as head motion, et cetera. There's dynamic connectivity that's driving a lot of the variance in connectivity values that might affect how we get better effects, because as we're bagging, we're sort of getting a better representation of that most dominant or central tendency of the data. So in conclusion, we see that bagging improves reproducibility, functional parcellation across scans, sessions, samples, studies, scanners, clustering techniques, and spatial constraint approaches. I think I'm basically out of time, so I'm not gonna really jump into too much of the of my Nilearn stuff, but I just wanted to say that I have really enjoyed teaching Nilearn and Python to people in Brooklyn. At this place, they have these classes and they have this lab. It's like a wet lab, but I'm like one of the people who does more of like the hacking tech, side. And I teach these classes, Neurohacking 101 and 201. And they're most of the people who attend are like, a really big range of ages, different professions, et cetera. But everyone in the public is just so fascinated about the brain. So showing them using, the great documentation and tutorials that Nilearn and Scikit-learn have available, we can actually, sort of show them how to download data and analyze it on their own laptops, which people get very excited about. And obviously I teach them all about all the really exciting projects that are happening in the open science movement, and they're all very excited to learn about them as well. So with that, I'll end my screen share. Thank you guys very much. How should I, there we go. Can I close the screen share?
It's all good now, so thank you so much, Ivan. Necessary groundwork is with PyBask the community a big favor. We have questions. If we ask questions, for those already mentioned, but if you, we got one for
Pierre, you're breaking up for me. Should I answer some questions, or are we?
oh,
cut
I'm
it?
sorry. Please, if
Okay. Okay, I think I heard you say to answer some questions. So I see, given bagging improves reliability more strongly than scan duration, what minimum duration would you recommend? If one were to be using BAST, and when would you expect to see diminishing returns when increasing scan duration? Based on what we see in the paper, I think 15 minutes to 20 minutes would be like the sort of the place where bagging hits improvements in reproducibility across the most conditions. But we also show that bagging just at the individual level, so not using any bagging in the time series, but just bagging the across individuals also shows improvement in reproducibility of the population, which suggests that maybe with even less data, you can get improvements regardless of how much actual scan time you have. I see, I believe you said that bagging with shorter scans produces more reproducible parcellations than using longer scans. Could this be because bagging duplicates the shorter scans, making it an apparently more resting, steady state? Easier to have steady mental state for a short period than a long period of time. Yeah, that's an interesting, that's an interesting point. The idea that like variability, I guess it kind of that kind of, you know, brings up the question of like, on what, and I'm not an expert on dynamics, functional dynamics, but like on what time scale are we expecting to see fluctuations in the major patterns here? If it were to be that exact thing that you're suggesting, then we would also expect to see 3 minutes and 5 minutes of data always doing better as well, which we don't. So it does seem like you need at least a minimum amount of data, like signal, so to speak, in order for bagging to have a positive effect. And that kind of also supports what we know from the bagging literature more broadly, that like, if you have a little data, bagging is actually gonna hurt you because your variability is gonna be too great. So that, it could be something like that that's happening in those low amounts of data as well. I see, which statistical metadata would you use to evaluate how many resampled data sets are appropriate for bagging aggregation? Well, you see in our analysis that we see even 10, right? So that's one of the cool things. Even if you resample the data 10 times only and aggregate those 10, do you see improvements in the reproducibility? So that could be also you know, something to consider, that we might not need that many actually to see an effect. And hopefully one day maybe we can put some kind of bagging thing into Nilearn or something like that. That would be really cool. In your opinion, could within subject bagging be used to drive more reliable functional connectivity matrices, or is it specific to parcellations? Yes, that is, I think that is a reasonable takeaway from this. I mean, if you are getting a more, and that is something that I have looked at a little bit, but I haven't really delved into it, but I do believe that that would be the case, that bagging should also lead to more reproducible functional connectivity matrices, because the thing that the clustering is actually using is just a functional connectivity matrix of the supervoxels. So if that underlying thing thing is more reproducible, then the clustering would be more reproducible as well. But it could also be that it's the variability in how the clustering works itself that is the problem. And so it's the part about aggregating across multiple clusterings that we're getting the improvement in reproducibility. So it's an important question. I do hope that people look at it, and we are looking at that exact point as well. Okay, I think that about answered that. Unless I missed one, yeah. Okay, here. Thank you so much. I think I've gone all my time. I'm sorry to prevent from going to lunch. I think that's lunchtime, right? It's morning for me, so I'm having coffee. So is that it here?
Like cutting it out, it's like, it's just cutting. But you get active for the space to see how basically sense Nilearn and this great US to so basically our session invokes figures against answering reactionally for joining and actions. Now get some lunch and make space in all about science or time as well. If we would see you in 1 hour, that is for the next session. And again, folks, and